you just send me an email and we'll work it out. Okay, let's start. So let's talk about what makes something alive. Okay. Do you see? Okay, good. It's working. Okay, so first thing, all life is obsessed with replication. Okay, that's the main purpose, basically, in essence, of everything that's alive. Its purpose is to replicate its genes. This is called the selfish gene theory. Okay, so that's one purpose of everything that's alive. So I'm defining for you things, what makes them alive. Okay, and one thing is they're obsessed with replicating their own blueprint. Okay, so the selfish gene theory is a gene-centered view of life, focuses on the gene. So that means all natural selection, all life, all forces apply to the gene. And basically an organism is essentially just a machine whose purpose is to replicate its genes. Okay, that's selfish gene theory. And that is the reason I'm telling you that is because that's one key component of what makes things alive. I'm testing this. Okay. Next thing is entropy. Okay, so all things are organized. Who's this? Hi. Oh, hey, you got in.
Yeah, well, trying to. So
okay. Okay, no problem. I don't have an extra handout for you.
Okay.
I'm sorry. So the second thing that is characteristic of all life is they're organized, right? And if you've taken physics, the second law of thermodynamics is that all the universe goes from a state of organization to disorganization, and life's purpose is to disrupt that process. So it spends energy becoming organized. So that's the second characteristic of life is reverse entropy, high organization. 3, metabolism. So I'm talking about characteristics of things that are alive. I'm starting with the basics because if you want to build something that's alive, I want to teach you guys start with the key components, the building bricks you use to build things that are alive. Okay, so the third thing is all things have metabolism. Metabolism. Okay, so that means they have processes that can take energy and use energy. Okay, and they build inside the cell, they build things themselves. They build chemicals, they build molecules, and that goes back to the entropy. So in order to reverse the state of entropy in the universe and build complex molecules, you have to have energy, and you have to put energy into the system. So they have to have a way to take energy from their surroundings and use that to reverse the entropy of the universe. Okay, fourth thing, adaptation. Okay, so this means that when they replicate, they're passing their genes on, and there's changes, not necessarily, but there can be changes in the next generation that allow them to adapt. So they change. They can adapt to their environment over generations through evolution, and they can also adapt to their environment through, in terms of humans, we can think, we can change our behavior. Lots of organisms can change their behavior. They can sense chemicals. They can change the structures of their surfaces. So they can change the organization of themselves and adapt. Okay, so these are 4 basic characteristics of things that are alive. Okay, so do you think computers are alive? No, computers are not alive because computers don't replicate at this point. But do computers use energy? Yes, they have a way of taking in energy when you plug them in. Do they adapt? You could program a program to adapt. Do they build things? You could program a program to build things and organize data. So computers are like have 2, 3, 4, but they don't replicate. So if they replicated, maybe we would consider them to be alive. Viruses. Okay, so are viruses alive? What? Yeah, we don't. It's in between because they fulfill all these things. They replicate. They organize themselves. They can adapt. They can evolve. Some of them will have things of metabolism, but they don't. They can't do it by themselves. So they can do all these things, but they can't do it by themselves. So this is kind of like question mark. Let's see what else is in here. Are you alive? Yes, obviously you are alive. Because you do all these things, right? So you know that you're alive. Okay, so now that we know the characteristics of things that are alive, we want to focus on a class of how we can build things that are alive, how we can change things that are alive. So let's talk about the structures of the cell. And I just want to make sure, what was the first figure that I put in DNA. Okay, so let's talk about structure. So the first thing we talk about the plasma membrane. Plasma membrane. Okay, so characteristics of all living life. Another thing that you note is that they are organized into compartments. Okay, and each of these compartments has a little wall. Okay, and it's usually, so if you talk about the cell membrane, it's the bilayer. Okay, and in this are lipids. Okay, and this is literally a wall. Things don't just go through and out haphazard. This is a wall. Okay, so they're compartmentalized. So the key component thing, if you're going to think about organizing or engineering biology, is you have to understand that these compartments are barriers and limitations. Okay, so if you want to say, if this is a eukaryotic cell and it has a nucleus and you want to engineer it to do something inside the nucleus, you have to understand that there is a barrier right there. There's a barrier called the nuclear envelope, and you have to program that into the way that you're going to manipulate the cell. You have to program a process of getting into the nucleus or getting out of the nucleus. Or if you're going to design a new drug that targets a particular cell, you have to design it in a way where you program it so that it can cross the barrier. Okay, so you need to always keep in mind when you are thinking about how to engineer organisms that there are barriers. And things don't just go where you hope they will go. You have to program them to go where you tell them. The cell is basically like a computer program. And if you do not tell a protein to go to the nucleus by programming its sequence properly, it will not go to the nucleus. So you have to understand this when you are engineering things. So this applies to, I'll just talk a little bit about my note system here. So the notes, if you see bold, it's like a keyword that I expect you to know what it is. And if you don't know what it is, I expect you to Wikipedia it, because most of these are pretty simple. If you haven't had them before, then send me an email and I'll tone it down a little bit. But I like to start off fast paced. And you let me know if I'm going too fast or if I'm talking about things that you have not heard about yet. So if I talk about something you never heard of and you're confused, you say, stop, talk about that. Okay? Is that clear? Okay, good. Let's see. Okay, so I'm talking about barriers, talking about how you engineer things. So let's talk about one like secretion systems. So I study bacteria that live inside of cells. So if this is a little bacteria, it lives inside of a host cell, okay? And sometimes these are called symbionts. And sometimes we want to engineer them to have an effect on the host, which might be a plant, might be an animal, but we want to engineer the bacteria to do something. So we have to recognize that the bacteria often have a lipid bilayer themselves. They have a cell wall, and oftentimes they live in a vacuole. So if we want this bacteria to pump something out into the host, we have to design it with secretion systems. We have to understand the secretion systems. So these are going to be systems that are built into the membranes. And we have to program whatever we want that bacteria to do to be compatible with this secretion system so it can secrete things out into the host. Okay, does that make sense? If we are thinking of a plant, if you want to engineer a plant, okay, you got a plant, you got a cell wall, and let's say this is in a leaf. Nice leaf. You want to engineer that plant to kill insects, okay? Well, you got to tell us. So let's say you give it a gene that is a peptide that will kill insects when they eat it. You better program that peptide to be pumped out through some kind of a secretion system to get outside the leaf, so that maybe you want it to get into the air, so maybe you want it to be a pheromone or something. You want it to attract an insect or something like that. You have to program that in, okay? So you got to keep that stuff in mind. Let's see. Sometimes some of these things I'll put in here and I think it's worth skipping. So some of these things I'm going to skip. So NPC is like nuclear pore complex. That's going to be like a way that you program things to get into the nucleus. There's a little hole called the nuclear pore complex, and you got to program stuff to go through that. And there's going to be nuclear localization signals. So like I'm talking about, you got to program all this stuff to happen. The way that you program it is there are signals for this kind of stuff, and they're encoded in the gene. So with the base pairs, and the base pairs code for peptides. And oftentimes there's special peptides which are recognized as signals to say, go to the nucleus. So an NLS called nuclear localization signal. Okay. So these things you got to keep in mind, you got to program these things. And we'll talk about these more in detail when we come across them in the literature. I'm just kind of giving you like a broad overview of all the things you have to consider if you're going to consider biotechnology and engineering organisms to do what you want them to do. Cell wall, okay, cell wall. Cell wall. Not only are many cells coated with a membrane, but many cells are coated with an actual wall that's made of tough peptidoglycan, which cross-links. Okay, and it's a hard wall. So not only do you have to program stuff to get across the membrane, you got to program it to get across the cell wall. Some bacteria have cell walls. Some bacteria do not have cell walls. Some plants have cell walls. Some yeast or fungi have cell walls. Some don't. Animals do not have cell walls. So you got to keep all this stuff in mind. What's in membranes? Okay, so let's talk about membranes more. What's in membranes? Okay, so the membranes are often the interface at which an organism will interact with its environment. So what kinds of things can you think of that would be good to put in a membrane?
Like
hormone receptors. Receptors, fantastic. So a receptor is going to be something that receives a signal, a chemical, ligand, and then it does something inside the cell and causes something to turn on or causes something to turn off. So it's a way through which the cell can adapt. Characteristic of life. Very good. These are things that you would put inside of cell membranes. In bacteria, there are receptors. There are what are called outer membrane proteins. And outer membrane proteins oftentimes do many things. Sometimes they act as receptors. Sometimes they feel around and hide themselves. Feel around and hide themselves from immune systems. So if you want to engineer bacteria, you got to consider that the plant or the host might actually have an immune system, right? And that immune system is going to be programmed to kill that bacteria if it doesn't recognize it. So maybe you need to change the structure of the surface so that it is no longer recognized by the immune system, right? So all these things we need to keep in mind. Porins, very good. Porins. So like I said, the cell wall or the membrane is like a wall. And so a porin would be like a channel, which allows passage of certain things to go in and certain out. And sometimes it's under special conditions. So sometimes it's regulated and sometimes it's more open or not. All these things you need to be aware of what they are. And all these things can be hacked and reprogrammed to do what you want them to do by adjusting the base pairs of the gene, thereby changing the structure of the protein, thereby changing the function. Okay. Let's see where we at now. Organelles. You guys should know about organelles like mitochondria, ER, Golgi, lysosomes. Like you've heard that stuff, right? Okay. You should not expecting that you know what that stuff is. And all that stuff you got to keep in mind, because if you want to adjust the function of metabolism, you might want to program a protein to go to the mitochondria, and you have to program a signal to go to the mitochondria. You got to know that stuff. You got to worry about, so when cells do endocytosis, so let's say they take in energy from the outside, and let's say you inject some kind of hormone into the bloodstream of an organism, and you expect it's going to get endocytosed. You got to understand that the process of endocytosis is to go to the lysosome, and the function of the lysosome is to destroy things and eat them. So you got to program whatever chemical you want to have an effect in the cell to escape the lysosomal pathway, right? You got to program that to get out. So this is just reviewing everything that you guys should have learned about biology. And now you have to understand the context of if we want to change that. You got to keep all that stuff in mind. Let's see. Okay, so you know what cytoplasm is. Cytoplasm is the liquid in a cell. You should know that. And then there'd be nucleoplasm, which is the plasm in the nucleus. Okay, so we know that. Organization beyond the cell. Okay, so you also have to have a broader understanding of when you think about biotechnology, you are not just thinking about the organism. Okay, that's like one thing. And within the organism, you're thinking about the cell, but you also have to think about the organism as a whole, right? If you program a brain cell, you have to think about how that's going to have an impact on the whole organism. But that's not the whole story. Organisms, and maybe you may or may not have learned this yet, but actually, like the human body, all the cells in the human body are not human. Most of them are bacteria, okay? So most of the stuff that's living in your body is bacteria, okay? So a lot of the things that make up you are not you. And you got to understand that that's true for most organisms. So most organisms like plants, there's a lot of organisms, bacteria, nematodes, things that live in the dirt, things that live in the soil, things that live in the roots. There's a lot of organisms that make that plant allowed to survive. And you might not know about those interactions, but you got to be aware that those kinds of things exist, okay? And this is the concept called the holobiont, which is the holobiont is an organism is not just an organism, it's an organism plus all the other associated organisms that live with it that allow it to survive and thrive and adapt. That's what's the holobiont. You got to understand that if you're talking about biotechnology, because maybe you don't need to engineer the plant. Maybe you just need to engineer the bacteria that lives in its roots or something like that. Okay. You also got to understand the ecology. Okay. So it's not just organisms that we're thinking about. When we're talking about biotechnology, we're talking about taking new organisms and putting them into a new environment that they've never been in before. Okay. And that's going to cause downstream effects that we may or may not be able to measure. We may have no idea what's going to happen to the ecology when we start playing with nature, which is fun, but we got to have an idea of the kind of effects that can happen when we do that. Okay, and we got to at least be thinking about the ecological impact of the things that we're going to be doing. So there's different levels, and I encourage you guys to start thinking about different levels, not just the cell. And within the cell, actually, there's molecules, right? Most of the stuff in biotechnology that you're doing is you're engineering a specific molecule, right? And then that molecule changes the function of the cell. And then that cell changes the function of the whole. And then that whole changes the function of the holobiont. And then the holobiont changes the function of the ecology. And maybe you get more productivity in your agricultural field. Does that make sense? Yes? Okay, good. Am I going too fast? Is this good? Okay. Okay, now let's get into some of the basic biochemistry. Who has had biochemistry? So 3, you've had it. So 2 have not had biochemistry. Okay. So here's what I'll say about the biochemistry. Let's just look at this. If I expect you to know a structure, I will put it here. And it's easy enough to remember structures. And the structures that I force you to remember are not useless. You will use these for the rest of your life if you are interested in biotechnology. So the things that I tell you to memorize are super important. And I'm not just giving you random shit that I'm like, you need to know, but you do need to know this. So like, for instance, if I tell you to memorize the structure of DNTP, the reason I tell you to memorize these structures is because this is what you actually put in your PCR reactions. And my policy is you don't ever put something in a test tube if you don't know what it is or what it's doing. That's a way to shoot yourself in the foot. You got to understand the things that you're working with, okay? So I don't give you like thousands of structures. I just give you a couple things. So like a DNTP, you want to know what that is. Here's a DNTP. This is the same thing. So it's not like I'm giving you 1,000 different structures. And I'll talk about these things as we go through the lecture. How much time do I have? I have 30 minutes. Good. Okay. So I can slow down a bit. Okay, good. Okay, nucleic acids. So when we're building biology, there's 4 molecules that make up biology. Okay, there's 4 building blocks. There's 4 types of chemicals that we're working with. The first is nucleic acids. Nucleic acids. Memorize this for the rest of your life. If you didn't learn this in biology, you need to memorize this. 4 types of molecules: nucleic acids, proteins, carbohydrates, which are sugars. And what's the last one? Lipids. Okay. So these are different things, okay? You find sugars in nucleic acids. That's the one caveat. But essentially, these are different things, okay? And there are people who specialize in studying 1, 2, 3, or 4, okay? And 1, 2, 3, or 4 are going to have different methods. They're going to have different data. They're going to have different everything, okay? And once you get beyond the undergraduate, you start to specialize in kind of like one of these things. And if you're good, you learn a little bit more. You broaden yourself out. But I just want you to know that there's 4 different types of molecules. And people spend their lives studying a specific set of things. But it's good to have a global understanding that there's all these different molecules, these 4 different types, and you want to understand how they make up the cell. Okay, so let's start with the nucleic acids. Okay, so nucleic acids are made up of DNTPs. So let's go to that. Let's look at that. DNTP. Okay, here's a DNTP molecule. There are 3 phosphates right here. 1, 2, 3. Okay, this is deoxy. So there's 2 different types of nucleic acids, right? There's RNA and there's DNA. What's the difference between RNA and DNA? DNA is deoxy, RNA is oxy. So in RNA, you would find an OH here, okay? But if we're using bases for DNA, DNTPs, there is a hydrogen here, no oxygen, okay? So that's what D stands for in DNTP. TP stands for triphosphate. N stands for this. I'm blanking on what the actual name is. Skip that. That just means you don't need to know it. If I don't know it, you don't need to know it. But this is a sugar, 5-carbon sugar. And it's important that you take a look at and note that there are these numbers. 1, 2, 3, 4, 5. Okay, 5 carbons. And this is important in the organization of DNA. And you want to know how DNA is put together, because if you do a PCR reaction, you are literally putting together DNA. So you want to know the building blocks, because if you mix up a PCR reaction, so what's attached to carbon 1 is a base, right? You've heard of A's, T's, G's, and C's. So that gets attached right here. And DNA then is a polymer of these DNTPs. So let's look at how that forms, okay? So if you take DNTPs, here's a DNTP right here. It's got a guanine R section right here, okay? When this thing gets put on to a polymer of DNA, 2 of these phosphates get lost, and this phosphate gets attached to right here. Now, the functional consequence of this is that there's what's called a negative phosphate backbone, okay? You need to know this. Why is this important? This is important because if you ever work with DNA, DNA is negatively charged. So everything we do with DNA relies upon the fact that it's negatively charged. The reason that it's negatively charged is because it has a phosphate backbone. Does that make sense? Yes? Okay, very good. Let's see, got that. Adenine, thymine, guanine, cytosine. I don't think I'm forcing you to memorize those, but know what they are and know that A binds to T and G binds to C. Oh, here's what I want you to know about A, T, G, C. Here's exactly what I want you to know. You need to know that G's and C's bind with 3 hydrogen bonds. Even better if you know the structure, but I won't force you to know the structure. But I want you to know that they bind with 3 hydrogen bonds, okay? A's and T's bind with 2 hydrogen bonds. Why is this important for building stuff? Well, when you build stuff, everything that you build is based off PCR, which is a way to replicate DNA, okay? And when you do PCR, you have to build primers. And a part of that process is the temperature at which your primers anneal. And the temperature at which your primers anneal is regulated by how many G's or C's you have and how many A's or T's you have. And that is encoded in exactly the sum of the hydrogen bonds. So which one of these is going to bind tighter? G's and C's or A and T? G and C, exactly. So when you design PCR primers, you do what's called the GC clamp, and you put a G or a C on the ends of your primer, and you can fill in whatever you want in between, but you want the ends to bind tight, okay? So this is when I highlight it yellow, it means I'm telling you, learn this because it's important for building stuff. And it's important for building stuff because of the GC clamp when you design PCR primers. Let's see. Okay, we are past get back to this. We are past nucleic acids. So I expect you to understand the basic biochemistry of nucleic acids, okay? Central dogma, nucleic acids code for protein. Okay, so let's talk about protein, which was the second type of molecule that we're going to be talking about. Okay, so proteins. So another pattern that you should be seeing. So if you're good at biology, you understand the structural patterns underneath everything. The pattern that you're seeing is that things that you engineer are made of building blocks, okay? And they're literally like little Legos, and each of these little Legos has a set function, okay? That's why when I made the poster, I made a picture proline, which was basically analogous to a Lego, okay? Because when we're building, we ought to understand the pieces that we're using, okay? So proteins are made of what? Proteins are polymers of amino acids, okay? Very good. Now there are 20 canonical amino acids. And in if you look on the website, I have a quote unquote combined PDF, which has all the lecture notes, the reading assignment. And in this lecture, I included a cheat sheet of amino acids. You want to memorize your amino acids, and I will expect you from now on to know the chemical structures of your amino acids. And I'm not being mean, I'm helping you out for the rest of your life, because if you understand the structural functionality of amino acids, you understand how to engineer and build proteins, okay? So we can start slow, and maybe I'll put like 10 amino acids on the first test, but know that you're gonna have to know your amino acids. And for the rest of the class, I'm gonna periodically draw this amino acid on a test, okay? So know your amino acids and start drawing them out. There's 20 of them, it's easy to remember, and you will thank me for the rest of your life for memorizing these. And it's fun because you're learning the building blocks, okay? So let's talk about amino acid, and go over the amino acids. So I guess it's saying that I should spend some time pulling up that sheet, I guess. I still got 20 minutes. So let me just pull this up as a demonstration of the combined PDF. So the combined PDF is going to have your lecture notes, the structures I want you to memorize. Here's a nice attachment, okay? So here is the sheet that you can use to help you memorize. So what you want to learn about the amino acids, okay, is that the amino acids have a basic rudimentary structure. Let me pull that up. Take notes on it. I'm still learning this new computer. Okay, here's what I want you to know. So let's start here. Let's start with the most simple amino acid. This is the most simple way to draw an amino acid. If you can't draw it, you don't understand it. I expect you to be able to draw it. And this is the most simple thing, okay? This is easy. You want to know how to draw this, okay? There's this carbon in the middle. Carbons have 4 bonds that they can make. On one side is a nitrogen. On the other side is a carboxylic acid. And in glycine, in the case of glycine, okay, so all amino acids, all of the 20 have 1 hydrogen, and then they have what's called the R group. The R group is a random variable. The random variables are different groups, okay? So let's say you have your R group here. The different groups, okay, are going to encode different functionalities. Functionalities, okay? So for instance, let's talk about glycine. Glycine's R group is a hydrogen. For those who have taken biochemistry, why does this make glycine unique?
It's the only one that doesn't have a carbon attached to it, or carbon within the R group.
Very good. Why does that change the function of glycine? What happens if you put glycine in a protein? No, okay, so glycine, so what she said is right, that there's no carbon here, okay? And the carbons all restrict the rotation. So if you put glycine in a protein, glycine allows proteins to freely rotate. So if you put glycine in the middle of a protein, you programmed it to have a hinge. Does that make sense? So you can use glycine as a hinge, okay? So this is true for all the amino acids. All the amino acids, if you look at, bam, look at this thing. All the amino acids have unique R groups, okay? And you want to learn what these things do. And at the end of the lecture, I'll go through some notes about what makes these different building blocks unique. But for instance, glycine, which is the tiniest, allows free rotation because that R group has no carbon. Okay? If you look at proline, where's proline? Proline is my example for the exact opposite. Okay? So if you study proline, proline puts a kink in proteins. Okay? And it does not, I'm going to spell stuff wrong the whole time, and I don't care about spelling. So if you do, whatever, don't get upset if I spell stuff wrong. Just random note. Proline, okay, so proline, if you program proline into a protein, it literally puts a programmed bend in the protein, and it is unchangeable. And I say that with a caveat. You can change it if you have an enzyme, which calls a peptidoprolyl cis-trans isomerase, which can flip prolines. But basically, if you can program proteins to rotate, to be free, or you can program them to be stiff and structured, right? And if you put a bunch of prolines in there, you might do that. If you put a bunch of glycines in there, you're gonna program it to be freely rotating. Does that make sense? So all the amino acids have special properties, special unique things about them. So there's charged ones. Let me just look and see where we're at in the notes. Okay, so there's charged ones. In the charge, there's going to be positive ones. There's going to be negative ones. I expect you to know what those are. What's going to be a functional consequence of the positive ones? Well, if you look at the positive ones, which is going to be lysine, arginine, they're charged. So imagine you want to program a protein to bind DNA. You want to program a transcription factor. It's going to turn a gene on. Is it going to work if you put a bunch of negative amino acids in that protein? No, because DNA has a phosphate backbone, which is negative, okay? So the regions that bind DNA are often filled with lysines and arginines, which are positive, and then you can get these interactions with DNA. Now, there's caveats. You can program things to grab ions, like a zinc. You hear about the zinc finger proteins. They could maybe grab a zinc, and then maybe the zinc has your positive charge, okay? So there's caveats. But a lot of the proteins that bind DNA, and if you want something to bind DNA, are going to have these regions that are stacked with charge so that they can interact with the negative phosphate backbone. Note and go over amino acids. What else is there? You want to know that there are small and large amino acids, okay? And if you're talking about enzymes, so enzymes are proteins. If proteins is the higher hierarchy, a subset of proteins are enzymes. Enzymes are the verbs in cells. They do things, okay? Enzymes all are going to have a catalytic site, which is the actual molecules or the chemical, atoms. The atoms that are going to be the site that causes that biochemistry to happen, okay? And if you stack that area with a bunch of big amino acids, sometimes you can what's called occlude the active site, right? So if you want something to happen at a key cysteine and you stack a bunch of things around it that are massive, sometimes they can cover up that cysteine and so it's not active. If you put a bunch of small amino acids there, maybe you're now exposing that key cysteine and now maybe you've made a constitutively active or on enzyme. Does that make sense? So understanding the building blocks has a huge impact in designing proteins. Let's see, there's small, there's large, there's charged, there's hydrophilic, hydrophobic. There's a bunch of stuff. It's all on the sheet. The key things that I think about all the time are charge, the glycine, which is a free rotating. I'll go through the ones that I think about all the time at the end. Let's see, modifications. Okay. The cell is not static. Cell, not static. It's always changing. It's always doing different things. It's always changing. Proteins are not static. Proteins, not static. If you have an enzyme, most enzymes can be turned on or off, okay? And this can be programmed into the amino acids. If you program certain amino acids can receive modifications, which are going to change the structure, okay? And that's going to change the status of the enzyme, whether it's on or off or in between. Biology is not black and white. It can be a mix. Okay, so you want to just be aware that in the cell you make a protein. You have no idea what its modification status is. So you program a plant cell to make an enzyme. It doesn't work. Oh, you quit your job. You're sorry. Well, maybe you should actually look at the modifications first. Maybe you worried that maybe your enzyme got methylated and now it's shut off. Okay. So there's a lot of things that can happen when you start fiddling with the cells, and you got to understand how the cell itself fiddles with the biology of its enzymes. Okay. So they can receive modifications. Modifications that you should just be aware of, that you should know. And there's many, many more than this. I'm just listing some. Okay. So phosphorylation. So if something gets phosphorylated, phosphate is negative. It's going to change the charge status of some particular atom. And sometimes this causes things to change their structural conformation and they turn on. Sometimes it shuts things off. But phosphorylation can cause big things to happen. Phosphorylation is done by kinases. Is that correct? Yes, that's correct. And I'm trying to think of what the opposite is. Everything in biology has an opposite. So kinase is like what does the phosphorylation. There's something that takes it off.
The phosphorylase?
Sure. I think that's actually what I was thinking in my brain. But for some reason I thought like it's got to be something more complex than that. Dephosphorylase. But yes, there's always a opposite. So there's other proteins, other verbs that turn things off. Okay, there is ubiquitin. Phosphorylation is a little molecule with a couple atoms. Ubiquitin is a protein. So you can actually take proteins and attach proteins to other proteins. Okay. And ubiquitin in the cell has many different functions. Sometimes proteins are like machines. And if you have a machine that's too old, it's going to start messing up the function of the cell. And you got to have a way to get rid of it. So if you have a machine protein that's too old, ubiquitin will get attached to lysine residues, and you'll get what are called polyubiquitin chains. And if you have 7, approximately, it will be recognized by the proteasome, which is a big macromolecule machine whose function is to recognize ubiquitin and take this thing and degrade it and reuse the components. Okay? So ubiquitin is a modification that you can find on your proteins. And all ubiquitins can get added through, you don't need to memorize this. I'm just telling you, I'm emphasizing the point that there are cycles of on and off. And for ubiquitin, there is a cascade of enzymes called E1, E2, E3, which add ubiquitins. And there are what are called dubs, which are what I study, which take ubiquitin off. Okay? So there's always an on, always an off with these modifications. There's methylation. DNA can be methylated. Proteins can be methylated, okay? And all these things are adding groups that can change the structure of proteins. You would just be aware that these things exist. These modifications exist. Let's see. We talked about that. Protein structure. So we are going to be building proteins. That's what biotechnology is. That's what it comes down to. Building protein, I don't know why I put P, protein structure. Okay, so you want to know that there's basic levels of protein structure. There's primary, secondary, tertiary, quaternary. Primary is what?
Amino acid sequence.
Yes, the direct amino acid sequence. Okay, and the primary structure is going to dictate the secondary structure, which is what? Yes, what is that?
Folding.
Not yet. Well, sort of. There are alpha helix and beta sheets. Who's heard of this? Okay, so you've heard of this before. Okay. So let's go to our note. I just want you to understand, like, you don't need to memorize, like, oh my God, look at all these hydrogen bonds. You don't need to know that. But, like, know that if you string together amino acids, you can build helices, which are called alpha helixes, okay? Okay, so you can build these alpha helices, okay? And the other structural conformation is you can build these beta sheets, okay? So secondary structure of proteins Alpha helices, beta sheets. I had primary, secondary, tertiary, quaternary. Secondary structure is the arrangement of alpha helices, and beta sheets. And that was dictated by the amino acid string. Okay, so you make string of beads of amino acids. Those amino acids fold into alpha helices, beta sheets. Tertiary structure then is the global conformation of the alpha helices and the beta sheets. So you get like the final folding. So where I really think of folding is tertiary structure. So, okay, you get the final folding. So the final structure of a protein is going to be at level 3. And then 4, what's quaternary structure? Subunits, interactions with other proteins. So like I said, you build machines when you build proteins. And machines have many different components. And the way in which these components interact is quaternary structure. And that can be regulated by the specific amino acids all the way down to the primary structure. So if you put a cysteine, cysteines can form disulfide bonds, right? And that can link proteins together that can never be broken unless you reduce them with a reducing agent. Okay. Some of these things I say fast, but I say fast because I know we're going to talk about it more later. And this is just the first lecture. So again, I'm just giving you the broad overview. Okay. So does this end at 8:50 or 9? 8:50. Okay. Good. This has been a perfect lecture. Oh, I'm going to have to erase now because. Okay. So let's talk about some unique hacks in my experience that you want to know, you want to be aware of about the amino acids. Most of the stuff that, I don't know if this is true. Again, this is going to depend on your specialty, so I hate saying this. But I'm going to say it. Most of the stuff that you work with in biotechnology is going to be dealing with nucleotides and proteins. Unless you're like specializing in, or like if you do something special, or maybe it's just that I do something special and I do nucleotides and proteins. But basically, we talked about the 4 molecules, and I'm going to tell you hacks about the amino acids. So this is just going to be relevant to 1 and 2. Nucleic acids, which encode these amino acids, and the amino acids to make proteins. So I'm not going to give you a bunch of hacks on lipids or sugars, and I know there are those because I don't know any. But I'm going to tell you the hacks of amino acids, and what we're going to focus on is building proteins. Okay, so glycine, right? Glycine is an amino acid that allows free rotation. Okay. The other side of that is if you have an alpha helix and you add a glycine here, right here, you can disrupt the structure of this alpha helix. So now maybe when you remake that protein, maybe you no longer have an alpha helix. No alpha helix if you put a glycine. So what this is called a helix breaker. And biology is not black and white. You can find helices that have glycines. But in general, glycine is considered a helix breaker. Okay. Alanine. Alanine, I think our group is CH3. Is that correct? Hi. And is that correct? It should be correct. It's small. Alanine is small. Alanine is also the token vanilla amino acid. So let's say you got a real fancy cysteine, which regulates an active site in a dub. Okay, this is what I worked on. You want to shut that enzyme off. You make a mutation that changes that cysteine to the token vanilla alanine. The reason you do that is because you don't want to put something in there that's wacky. You just want to disrupt that function. So you change it to alanine. Okay. So alanine is like the safe bet for like when you make a mutation and you don't want to like destroy the protein, you just want to like just shut it off. You put in an alanine, the token vanilla, the small one. Proline. Proline does the bent kink. Okay. Proline can be flipped that way if it interacts with an enzyme called the peptidoprolyl cistranslysomerase. But you want to know that it does this bent kink. As such, proline is also considered a helix breaker. Because if you put a bent kink in that helix, it's no longer a helix. Aspartic acid, glutamic acid. Those are your negatives. So you want to know your negatives. You want to know your positives, which are lysine and arginine. You want to know that there's this dichotomy. There's this positive, negative, positive dichotomy. So why is this important? Let's say you have a DNA binding protein X, and it's got a special site with a lysine. You want to make it no longer bind DNA. How do you do that? You switch it, make a mutation. You change it to a negatively charged amino acid. So you get a key protein, it's got a key lysine. You know this lysine is important for interaction with the major groove of DNA. You switch it out to a aspartic acid. In theory, it should no longer bind DNA, right? So here's how you can change a function of proteins. Now, this might be very, very important for a negative control in an experiment. Let's say you have an effect that you think is conferred by DNA binding, and you want a negative control. You have to build a protein that no longer binds DNA. So you have to make a mutation that changes that function, okay? So that's why this is important, that positive-negative dichotomy. Pull up a PDF for this. Aspartic acid. Some things, some amino acids, like serine, get phosphorylated. So here's phosphorylated serine. Phosphorylated serine often turns proteins on or off. Let's say you want to engineer a protein to be always on. You never even want to give it the possibility to shut off. You can switch that serine for what's called a phosphomimetic aspartic acid. That means the structure of that aspartic acid mimics phosphorylated serine. And as such, maybe you've now designed a protein that is always on or always off, depending on context. Does that make sense? That's the unique thing about aspartic acid. Histidine. Biotechnology. Greatest example of protein purification is insulin. We need to make insulin to help people with diabetes. How do we purify insulin? Well, we make it in E. coli. How do we get it out of E. coli? I don't actually know if insulin has a histidine tag, but this is an example of what you could do with a histidine tag. Histidines bind metal, and if you string 6 of them together, 6 histidines, the codons are cat, so you could do cat, bunch of cats. Codes for histidine. And if you have 6 histidines, his6, this binds nickel or cobalt. So you make a tube, you fill it with beads that are metal, that will bind histidine. We're out of time. I just had some other stuff. Lysine gets ubiquitinated. Serine threonine gets phosphorylated. Cysteine can form disulfide bonds, quaternary structure. You break these apart when you do SDS page and you use reducing agents. Methionine, we'll talk about next time. All proteins start with the methionine because it is encoded by the start codon, ATG. Valine is sometimes an alternative start codon. There's also lipids, which are polymers of carbon, and sugars, which are carbons in water. There's a general formula. Proteins, final thing that you want to know, proteins can get another modification of sugars, which is called glycosylation. So let's end there. Any questions?
Yes. The phosphomimetics.
Yes.
So if something's phosphorylated, it just gets the phosphate attached to it?
Correct. It gets attached to phosphate, attaches to the R group of the amino acid. Does that make sense?
Yeah.
So phosphomimetics don't have a phosphate group. It's just aspartic acid kind of looks chemically like a phosphate attachment. Good? Great work. Do the reading. See you guys Friday.